Welcome to another episode of I Am Nano.
Putting the I in I Am Nano. I'm your host, Irfani.
And I am your other host, Monica.
And today we will be talking about nanopores.
Ooh, fancy nanopores. So it's not like the pores we have on our faces, though, right?
No. Not those ones. Those ones are pretty large. You can see them with our eyes.
Yeah, mine are anyway.
Oh man, I wish we have small pores, but unfortunately, that's not what we have on our face.
Yeah, for sure.
Pores are essentially small openings, right? And today we're going to be talking about an especially small pore, nanopores.
So we have pores in our cells, like the nuclear pore in our cell's nucleus. So going back to maybe our undergraduate biology classes, very nice, very interesting. And they allow certain molecules and ions to move in and out of the nucleus. They are important for various biological processes.
Right. And they are essentially a form of pathway allowing things to move from one side to another. So think of them, like gates or doggy doors.
That's great.
Yeah. Just like, how some of them are designed for small dogs and others for larger dogs. So pores can also be selective.
Awesome. And while, yes, there are natural pores that occur, today we'll be talking about pores made from DNA.
Man, DNA. I feel like we talk about DNA so often. We previously discussed how they are used to store information, used as sensors, and even as a scaffold for Iron Man's suit.
Yes, very awesome previous discussions that we've had. And, DNA is so very versatile, and it is a naturally occurring nanomaterial. Maybe that has something to do with it.
Yeah, and their properties are, amazing, so they can be applied for so many things.
Yeah.
So in today's paper, published very recently in ACS Nano, scientists from the UK, Netherlands, and Germany explored the transport principles and mechanism of DNA nanopores.
Interesting. And as we may know, complementary DNA strands bond together to form a double-stranded DNA. The DNA nanopore is composed of 6 complementary DNA strands to make a barrel-like formation. 3 cholesterol strands were linked between alternating DNA to anchor them on the lipid bilayer membrane. These nanopore lipid membranes were then placed over microcavities to evaluate the transport of various fluorescent dyes.
So how does this work? So the inner diameter of the nanopore is 1.8 nanometers. So only dyes that have a smaller hydrodynamic diameter are the ones that can pass through it, while others can't. And they used ATO 655 dye with a hydrodynamic diameter of 1.2 nanometers. And when compared to organ green dextran, which is another dye that is 3.7 nanometers in diameter, only the ATO 655 dye diffused out of the microcavities.
And now the ATO 655 has a -1 charge, so -1. But what about molecules with a higher negative charge? Well, they also evaluated the transport of the dye FAM or FAM that has a -3 charge and calcine that has a -4 charge. And what was discovered was that the rate of transport of both dyes was slower than that of ATO 655, with calcine having the slowest transport. Do you know why that is, Irfani?
Well, the DNAs have a negative charge, right? Because of the phosphate backbone. So if the dye has a higher negative charge, there will be electrostatic repulsion. Negative charge and negative charge repel and hate each other.
Right. And the molecular dynamic simulations in the study showed that the negatively charged dye, FAM, encountered a larger energy barrier during transport because of electrostatic repulsion, as you just described. ATO 655 did not face such a high energy barrier comparatively.
And think of energy barriers as like a hill that you have to face when you're running, So the higher the energy barrier, the higher the hill, the more energy you need to cross it, which, can sometimes slow you down.
Oh, certainly. Yeah. Definitely been encountered that before.
Yeah.
Yes, for sure. The larger the hill, the slower it is for me to cross, for sure. And furthermore to that, this nanopore's transport properties was evaluated to be quite comparable to a naturally occurring protein called alpha hemolysin. So it's pretty impressive that DNA-based nanopores can act similarly to already naturally made nanopores.
Dang, that's really cool. Yeah. And also, interestingly, they actually found that using lipids with a negative charge for the lipid bilayer helped to transport the negatively charged dye FAM. I mean, this is a little counterintuitive, right? Given that we just talked about how FAM transport is slower because of the negative repulsion. But it's actually thought that the nanopore is dilated because of the electrostatic repulsion between the DNA and the lipid.
Oh, very interesting. Lots of charged induced interactions going on with this for sure. It's kind of like also, maybe we can describe it as a couple on a date and they order two different desserts, but both have chocolate in them. And one of the persons really likes chocolate. The other doesn't really like chocolate at all in their dessert. So the one person can give the other person their chocolate, kind of opposites attracting here. And, both people are happy. But if they were both chocolate lovers and had these two different chocolate desserts, maybe we would go into negotiations or something.
Yeah.
So who tries what first and when, and who gets most of the chocolate. So it would be a much slower process.
That's very true. That's so true. That's a good, analogy there. I've been in that situation before, for sure. Well, anyways, yeah, DNA, man, they're so useful for so many things.
Yes, definitely. It's literally one of the building blocks of life, though,
yeah.
I would say.
Alrighty, that's all the Nano for today. Take care.
And stay curious.